Good afternoon, everyone. I'm Suzanne McGurn, President and Chief Executive Officer at CADIF. I'm pleased to be with you today, moderating today's webinar on blood plasma therapies for COVID-19. When this topic was picked a little while ago, little did we know how topical this would be in the news cycles both here and south of the border. As we get today's session underway, I'd like to begin by acknowledging that I'm speaking to you today from Ottawa, which is on the traditional unceded territory of the Algonquin and Anishinaabe people. I would like everyone to take a moment to reflect on the land from which you're participating in today's webinar. Based on some of the questions from our last webinar, there's a little bit of information we've moved to the front of the presentation. So just want to let everyone know that today's webinar is being recorded and will be available to you on CADIF's YouTube channel later this week. And yes, the way that the session today is set up is we will be hearing from our speakers for about 30 minutes, and then there will be an opportunity for questions at the end of that session. So about today's topic. 2 weeks ago, for those of you who also participated in the last webinar, we heard about the global push to develop a vaccine for COVID-19, which many experts see as the best hope for ending the pandemic. We certainly learned a lot, and there was still a lot of unanswered questions. But researchers and decision makers and the public are not putting all of their eggs in one basket, and many other treatment options are also being explored and researched. However, with the announcement just a couple of days ago that the U.S. Food and Drug Administration granted an emergency use authorization for the use of COVID-19 convalescent plasma to treat hospitalized patients, this century-old therapy was once again thrust into the spotlight. This is not a new thing. It has been used in the past to combat outbreaks of polio, measles, mumps, influenza. The question is, could it be one of the keys to ending our current COVID-19 pandemic? I'm pleased to tell you that today we have two of Canada's leading experts on blood and blood plasma therapies to guide us through the basics of what blood plasma therapy is, what we know so far, what about the clinical trials that are underway, and actually, and then interesting, how you can contribute to the study, a nationwide clinical trial on the use of convalescent plasma to treat COVID-19. I'd like to take a quick moment to introduce our speakers. Dr. Dana Devine, PhD professor at University of British Columbia and Chief Scientist at Canadian Blood Services, and Dr. Janine Callum, MD, professor at University of Toronto and a transfusion medicine specialist and hematologist at Sunnybrook Health Science Center. Our two renowned speakers are going to be tag teaming it for their presentation. They're going to move through these slides, which will be available on the YouTube presentation for about the next 30 minutes, and then we'll move into a Q&A. You will also note that you can submit questions at any time that the speakers are speaking, and by clicking the Q&A tab in the Zoom control bar. And if you want to direct your question to one of the speakers, it will assist me in directing the questions at the question and answer session. Include who you would like to answer the question if it's specific. If you do have any technical issues, use the Q&A tab to let us know what the problem is, and we will try and use them. The Q&A box will be monitored as the speakers are proceeding with their presentation. With that, I'd like to call on Dr. Dana Devine to get things started and lead us off to this interesting conversation today.
Great, thank you very much, Suzanne, and welcome everyone to this presentation. Dr. Callum and I would like to share with you some of the ongoing activity in trying to determine whether convalescent plasma is a useful therapy for COVID-19. Our affiliations are given on this slide. Can I have the next slide, please? And our relevant disclosures are listed here. And then the next slide, please. So what we're going to do is approach this presentation in the following way. I'm going to talk a little bit about the rationale for convalescent plasma, and then Dr. Callum will take over and talk a bit about what we actually know from some of the early clinical work, and she will talk in some detail about the Canadian trial, the Concord 1 trial. and really what the principles are behind the trial, how it's structured, and then talk a little bit about the status. And then I'll come back and talk about how we actually acquire convalescent plasma and some of the challenges we have around qualifying the product, et cetera. So next slide, please. So as Suzanne was mentioning, this is not a new idea. This has been around for quite a long time. Probably most relevant to our considerations for whether or not we would want to try this for COVID-19 are papers such as this paper that was published in Annals of Internal Medicine in 2006, which essentially several folks went all the way back to the original articles that were published during the time of the Spanish flu outbreak to look to see what the evidence looked like that convalescent plasma might be helpful in the treatment, at least in that Spanish flu outbreak. So this was a meta-analysis of papers that were published in the day, and as you can imagine, in the 1920s, the quality of clinical trials is probably not exactly what it is today. But even that said, they were able to demonstrate that there was a reduction in the case fatality rate among patients who received convalescent plasma compared to untreated controls, and that very importantly, that case fatality rate was lower, particularly if you were focused on patients who were receiving treatment in the early days of presentation. So generally for flu, it was about 4 days or less. If you waited and treated later, you had less of an effect from convalescent plasma. Go to the next slide, please. If we move away from influenza and consider what do we know about the treatment of coronaviruses with convalescent plasma, there was some convalescent plasma therapy used in the SARS-1 outbreak. There were about 80 patients treated in Hong Kong, and this little table here shows the results of that study. What they were looking at were good outcomes, meaning that the patient was discharged from hospital, and then poor outcomes were at day 22 after symptom onset, the person was either still hospitalized or had died. And so they were able to show that there were certain factors that were associated with improvement, and that one of them was the use of convalescent plasma. So they were able to show that the outcome with convalescent plasma was better in younger patients treated earlier in their disease course, who perhaps lacked their own antibody response. And I apologize for the telephone in the background. It's the curse of working from home. Could I have the next slide, please? So what's going on with convalescent plasma? Why are we thinking that this works? So convalescent plasma principle is one of passive immunity, that you're giving an antibody developed in a person who has had a disease to someone who is just coming down with the disease, whose own immune system has not yet developed antibody response, or at least an antibody response that's able to help them clear an infection. For the SARS-CoV-2 virus in particular, what the concept is that antibodies are able to interfere with the viral entry mechanism into cells. And so I think many of you probably well versed in how this virus gets into cells. It's using the ACE2 surface receptor of host cells to get itself taken inside the host cell, in which case its viral RNA is released and it goes on to reproduce. The relevant focus is really on the sort of little decorative flower-like structures on the surface of this virus, which is the spike protein, and how that spike protein interacts with the host receptor. So if I could have the next slide, please. In slightly less complicated cartoon, what the idea is for convalescent plasma is that you're introducing antibodies which are able to neutralize the virus and keep it from entering into the cell. And the way that we actually assess the plasma that we use for treatment is by actually doing testing to determine whether the plasma can neutralize virus. In other words, in an in vitro laboratory test, can we look to see whether plasma has in it antibodies that can keep a virus from infecting a cultured cell. And so we'll be talking a little bit about plaque reduction neutralization tests, or PRINT is the acronym for them, and you can determine whether your plasma is going to neutralize 50% or 90%, kind of the typical way that we do these assays. The next slide, please. So the other ways that antibodies may help is actually in a direct attack on the virus. And so particularly with IgM antibodies are strongly complement fixing. And so virus that is loose in serum would be attacked by IgM antibodies. The complement system would become activated, and you would either get direct virolysis, direct lysis of the virus by the complement proteins, or you would get the virus very well tagged for removal by macrophages. So in general, we are not looking at the IgM response when we are qualifying plasma for patient treatment, but there will be, in many cases, there will be some IgM in there. The other immune response that's mounted is an IgA response, and the IgA may actually turn out to be quite important in COVID infection because it's a mucosal immune response, and so the IgA levels may be important in actually preventing initial viral penetration. But the IgA response is not something that we're measuring in plasma because the IgA levels in plasma are quite low. So can I have the next slide, please? The other thing that's important to realize is that not everybody makes a robust antibody response to the COVID-19 virus once they've been infected, but yet they overcome the illness. And so that tells us that there clearly is a cellular component to the immune response to this virus, and that we may be seeing an antibody-dependent cellular cytotoxicity response, or a direct killing of infected cells by natural killer cells. And so the quality of the antibody for this type of response is less important because it's the immune cell that's actually mediating the killing. The next slide. Okay, at this point, I'm going to pass the baton over to Dr. Callum, and she's going to start talking about the clinical side of using convalescent plasma. Jeannie, over to you.
Okay, thanks, Dana. So when we started this conversations about this, doing a trial, we really wanted to make sure that we felt that there was clinical equipoise, that we really didn't know what the right answer was. And we still have that. So we had this low quality non-COVID-19 study suggesting a possible mortality benefit for influenza and SARS. And you can see there, I'm going to go into the data in more detail, but definitely low quality, small numbers of patients. And at the same time, that was balanced against known risks of getting a plasma transfusion. We know that most of the people we give plasma to, same for COVID-19 infected people, most of them are in an older age bracket. And so they're at risk of getting what we call TACO, or transfusion-associated circulatory overload, or heart failure, but that's a risk of 1 in 100. But they can also get other reactions like trolley, which is an antibody-mediated complication where antibodies from the donor attack the host lungs. And a risk of about 1 in 10,000, and also anaphylaxis. So those are the known risks, but there are also theoretical risks that we were worried about. Was it possible that there would be a risk of antibody-dependent enhancement of infection? Would these new antibodies help get the virus into the cells and actually worsen infection? Would be providing antibodies cause a cytokine reaction that would make patients sicker that were in the intensive care unit? And would it attenuate the long-term immunity and make them more likely to be reinfected later on? And so we thought that there were balance between these two, and that we were good to go ahead with a trial. Next slide. So this is the most recent systematic review. It's by one of our co-investigators on the CONQUER-1 trial, Dr. Philippe Began from the University of Montreal. And he pulled these 6 studies, 4 from influenza that were randomized trials, and then 2 non-randomized trials, 1 from SARS and 1 from Ebola, that met the inclusion criteria of being slightly higher quality. And you can see here, for mortality, there wasn't a lot of strong evidence that it was changing the mortality rate. Next slide. But what they did see was a suggestion that by giving convalescent plasma, the recipients had a shorter duration of hospital stay and shorter time on ventilation. So even if we don't change the death rate, these are really important outcomes for patients. So there was some suggestion that maybe this is beneficial. Next slide. The other data that we now have is about safety. So safety different from efficacy about whether it works. Is it actually just safe to give patients? And this came from the Mayo Clinic group that's doing a study of giving all patients convalescent plasma with no control group. So they're able to publish data on 5,000 patients with severe COVID-19 treated with convalescent plasma. And the number of serious adverse events in the first 4 hours after transfusion were very low, less than 1%. And 25 serious related adverse events out of those 5,000 patients, so very small. I have to say, when you read their methodology, which obviously is unpublished at this point, they don't describe what classification system they used for the transfusion reactions, whether they use the American classification system for transfusion reactions versus the International Society for Blood Transfusion. So I'm not clear whether or not this report will over or underestimate the risk of the convalescent plasma. For our trial, we're using the ISBT, the International Society for Blood Transfusion classification for transfusion reactions with adjudication. Next slide. So how about efficacy? So Dana talked about, in influenza, but what about the emerging data for COVID-19? So the same group from the Mayo Clinic, again, they're giving every patient convalescent plasma, reported in an unpeer-reviewed publication last week. 35,000 people treated with convalescent plasma, of which half were in the ICU, a quarter were on a ventilator, and not actually quite surprising. The most common age group was 40 to 59, and 60% of the patients were male. The 7-day mortality rate, which is really an odd thing to describe. If you're alive at 7 days, we care more that you're alive in 3 months or a year. But the 7-day mortality rate was better if you were treated within 3 days of your diagnosis, 12% versus 9%. And then similar findings at 30 days. So a little bit lower mortality rate if you were treated earlier rather than later. But look at the jump in the mortality rate from 7 days to 30 days. And so obviously highlighting it's really important that we have longer term outcomes in these patients. Next slide. What they did find in that publication was some suggestion there might be a dose response curve. So remember, this is a biological product. Not all donors are created equally. And so some of these donors had very high levels, immunoglobulin levels. Some had medium and some had low. And this was measured in what's called a signal to cutoff ratio by a test called the ortho, it's an ortho vitreous test that was used on these donors. And if you got a high titer, there was a suggestion that your mortality rate was lower. And it was lower at 7 days as well as at 30 days. And so it appears that maybe the quantity of the antibodies in the product might matter. Next slide. And the same effect was seen in this report that was just published out of Houston. This was 136 patients with 28-day outcome after being treated with convalescent plasma. Remember, everybody was treated in the study. Matched 251 patients that were not treated, but this was not randomized. And you can see there from these curves that patients that were treated within 3 days had a lower mortality rate. Patients that got a higher titer of product had a lower mortality rate. And if you were so lucky to get treated early and with a high antibody titer product, your outcomes were quite good. And so some suggestion that the antibody level matters. And Dana really showed her leadership to make sure that the Canadian product actually, right from the get-go, we selected high levels of antibodies for our donors. Next slide. These are the comparative trials that have compared convalescent plasma to no convalescent plasma. All but two trials were non-randomized studies, but again, suggesting a reduction in mortality rate with convalescent plasma. Next slide. So let me tell you a little bit about the Conquer-1 clinical trial that we're doing. That's been a lot of fun. So on March the 26th, we started planning this. And 3 of us came together. None of us are infectious disease experts or have any prior interest in convalescent plasma. Darnie Arnold is a big platelet researcher. Philip Began is an immunologist. And I'm a transfusion specialist who spends most of her time worrying about patients that are bleeding. But we redeployed all of our skills and efforts to come together as a team. We make a great team. We all have different kind of styles and qualities. And so we make a great team. Next slide. So our primary research question is, does the transfusion of convalescent plasma collected from donors who've recovered from COVID-19 and have detectable anti-SARS-CoV-2 antibodies reduce the risk of both intubation or death at 30 days in patients admitted to hospital with a COVID-19 respiratory illness compared to standard of care? Next slide. So these are our inclusion criteria for our patients. So they have to be 16 or older, admitted to hospital with a confirmed COVID-19 illness by PCR by a nasal swab, have to be on supplemental oxygen for COVID-19 respiratory illness, and we have to have ABO-compatible convalescent plasma available. That has not been a problem. We were concerned that it might be, but so far so good. They can be co-enrolled in other drug trials. Next slide. We have very limited exclusion criteria, so that the hope that this will be generalizable. So onset of respiratory symptoms more than 12 days before randomization, current or impending intubation, plasma is contraindicated for some reason, or there's a religious objection to transfusion, or a decision is in place for no active treatment. We're still enrolling patients who decline ventilation or ICU care, but otherwise want full care. Only the patients who are made immediately palliative are we excluding from the trial. And pregnancy is not a contraindication. Our first patient in the trial was pregnant. Next slide. So the patients are being allocated in a 2-to-1 allocation to COVID-19 convalescent plasma compared to standard of care. We did have a conversation about giving the standard of care arm the same volume of plasma, but we were concerned about sending nurses in to give these patients convalescent plasma and expose them more to COVID-19, as well as the risks of plasma, that there was no possible benefit to them, and the lack of a control substance we could use to mock up a plasma unit. They're getting 500 mils of ABO-compatible plasma. This is very different from what the FDA current recommendation is 200 ml, so it's a bit different. And they're infused over 4 hours and monitored for adverse events. Some patients are going to get 1 unit of 500 ml and others 2 250s, just depending on what stock's available for that patient, depending on how it was collected. Next slide. So assuming the baseline risk of intubation or death in hospital is about 30%, we estimated a sample size of 1,200 patients with an 80% power to detect a relative risk reduction of 25% between the convalescent plasma arms and the standard of care arms with a 2-to-1 randomization. So we're going to have, of the 1,200 patients, 800 patients will get convalescent plasma, 400 standard of care. To make this happen, we need about 600 donors. I think that because the varies of the amount of antibodies, so I'm hoping at the end of the day we have 400 that got high titer, 400 that got not high titer, so we'll be able to compare, does the dose matter? Next slide. So this is an investigational product. We can't use it on anyone else outside of a clinical trial, no matter how sick they are. There are 2 other clinical trials registered where we will be using this. That's the Conquer Kids, very similar to the Conquer-1 in adults, and another study for ICU patients called REMAP-CAP. Next slide. So our primary outcome is intubation or death at 30 days. But of course, this is a sick in-hospital patient population. So we're tracking classical outcomes from sort of an ICU sick respiratory population, including things like their time to intubation, vent-free days, length of stay in ICU and hospital, need for things like ECMO, which is a heart-lung bypass where the lungs that you cannot ventilate them, or renal replacement therapy, adverse events to transfusion, all of them. We're tracking every single transfusion reaction, no matter how minor, and then grade 3 and 4 serious adverse events. This is a very sick patient population. So for us to track every abnormal lab test would be impossible. So we're just going for things that really matter to patient outcomes. Next slide. So at the end of the day, we plan to have 60 randomization academic sites. That's what we're have built into our randomization structure. And that will include about 20 Ontario hospitals, 15 Quebec. It was very helpful that both Ontario and Quebec have a central research ethics board so that we could get a whole bunch of hospitals approved at the same time. There'll be 10 other pan-Canadian sites, and most of those sites are now open. We have 3 US sites in New York, and then we have a plan to bring up in the next month 10 sites in Israel and Brazil. And I've just included a map there. Those are some of the sites. There are now more sites active. I just don't have an up-to-date map. Next slide. So to date, so we started this journey on the 26th of March, and we've come so much faster. And that's because we really joined 3 large research teams to work together in concert to make this happen, along with the teams from the blood centers. So during that time period, we got a protocol approved. We have a steering committee, an independent data safety monitoring committee, a community engagement committee to make sure that all patient types are getting access to be in this trial. We have a logistics team for just distribution of the product to make sure it's where that ABO blood type of patient is. The protocol approved by both Health Canada and the FDA. The study approved by the research ethics boards at Canadian Blood Services, HEMA Quebec, and the New York Blood Center. Those blood centers together have collected over 1,500 doses. So they're far ahead of the game, and then was within a month of us. Even Dana Devine and her group got a neutralized assay up and running, and we got 58 hospital sites open, and 40 of our 80 randomization bursts have been taken, and we have secured funding to complete the trial. Next slide. And this is just our enrollment today. We have 88 patients as of this morning. You can see there, it's a good mix across the country of patients. And so it speaks to the importance of having a multi-site, because the disease is kind of popping up in one location, then going down, then popping up in another location. And you can see there that so far Ottawa is leading the fight. Next slide. And there are lots of other trials underway. I should have said, or they were other trials underway until Donald Trump announced making this available throughout the US. So you can see there are 53 trials that are in the same hospitalized severe ICU, kind of in competition to this, and including numerous multi-center trials. I'm not sure what's going to happen after yesterday. Some of those trials probably are no longer feasible, but time will tell. So with that, I'm going to hand it over, and Dana's going to talk about our success with blood donation and the blood donors.
Great, thanks, Jeannie. So I guess one of the questions that comes to mind is, where does all this convalescent plasma come from? And this is the plasma for this trial is coming from our fellow Canadians who are actually recovered from COVID-19 themselves and are willing to undergo a plasmapheresis procedure to donate their plasma. So this is a procedure where you're actually attached to a machine, and we withdraw the blood. We give you back your red cells, white cells, and platelets, and we keep the plasma. So a donor would donate between 500 milliliters and 750 milliliters of plasma, depending which type of equipment we actually are using for the collection. So the next slide, please. So I just wanted to say a few words about the antibody that we're collecting and how we test for it, and really some of the complications that we have there. So in a typical immune response, you have a short period of time where your immune system is just sort of recognizing that there's something foreign around. It's starting to mount an immune response. The education of your lymphocytes is occurring, and you're starting to produce antibodies. initially you make a fairly low level of not very strong antibody that is both IgM and IgG. And then as time goes on, you get more IgG produced, and that IgG starts to have a higher binding ability, so, or what we call avidity of the antibody. In most infections, you get a peak of the IgG production occurring about a month after you've had the infection. So your immune system sort of gets revved up and then keeps revving up for a while. That infection will clear, and then your immune response basically is like, yeah, okay, that one's done. We're waiting for the next one. And the level of antibody that you have in your plasma starts to decrease. And that can take place over the course of a few weeks, up to a couple of months. What we don't necessarily know for any given infection is whether your immunity is going to be lasting. So you'll know that, every 10 years we have to go back and get a tetanus shot again. And this is a reflection of the fact that your immune system isn't necessarily standing on guard for things it's seen before for an indefinite period. We're concerned about what we call neutralizing antibodies because we think that from a scientific perspective, those are most likely the active ingredient in convalescent plasma. And we know that of the donors that are being collected so far, or even patients that you test for these kinds of antibodies, about 2/3 of people make a pretty high titer neutralizing antibody at some point in their immune response to SARS-CoV-2. Have the next slide. So one of the things that we worry about a little bit is whether the virus, as it's moving around the globe and as time is going by, is this virus mutating? And does the mutation of the virus matter with how your test actually functions for the virus? So this is a question for which we don't actually have clear answers yet. We do know there's a couple of strains of virus out there. One is what we refer to as the European version, and the other is the Chinese version. And in Canada, we have a mix of the two. At the moment, it looks like the tests that we use to look for these viruses are picking up both types of viral infection in a fairly straightforward manner. But viruses do mutate, and so we are always concerned about making sure that we have a test that can still pick these up. And it may be that at some point one needs to have more than one type of test. Next slide, please. So if we go back for a moment to what are we looking for with these convalescent plasma donors? From the Canadian Blood Services perspective, what we've done is actually we want to know if someone has had a positive test for COVID. So these are generally people who have come down with an illness, have gone out into their provincial testing system and been tested for using an RT-PCR assay. We also will take as donors people who are presumptive positive. And what does that mean? That's somebody who say is in a close living situation with someone. They were both sick at the same time. The partner got tested and was positive for COVID-19, and the individual didn't bother to get tested, assuming that they had the same thing. So that's a presumptive positive, and we are accepting presumptive positive donors. For the trial, both Canadian Blood Services and our sister organization, Hema-Quebec, who operate the blood system in the province of Quebec, we have been collecting from confirmed and presumptive positive donors. All of these donors have to meet the qualifications for a regular plasma donor in both our organizations, and that's because this plasma is going to be transfused into people, and so all of the safety parameters that we have in place, both for the donor safety and for the recipient safety, must be met. So we ask that long list of rude questions. We ask all blood donors. We want our donors for plasma to be less than 67 years old, and donations are all tested for the usual list of transmissible disease markers, and those all have to be negative. The plasmapheresis procedure, as I was mentioning, returns all the cells to the donors, and we're collecting either 1 unit of 500 mils or 3 pouches of 250 mils each, which is why Dr. Callum was saying before that sometimes you might get plasma from 2 different donors depending where your 250 mil unit came from. Importantly, donors can donate plasma as frequently as once a week. That's the allowable frequency of plasma donation. And so people can come and make multiple donations during that time when their antibody levels are high, and they're useful for this trial. So initially for Canadian Blood Services, we've been seeking male donors or females with no history of pregnancy. And this relates to one of the complications Dr. Callum had mentioned, of the possibility that someone, particularly female donors who have been pregnant, will often have antibodies against white blood cells that they made in response to antigens that were on the fetus white blood cells. And those, if they get into the wrong person at the wrong time, can cause, really a catastrophic transfusion complication called transfusion-related acute lung injury. So we felt that this was the worst thing you could possibly do to somebody who was hospitalized with a bad lung ailment. And so right up front, we basically have made the decision not to collect plasma from that cohort of potential donors, at least initially. We may be doing that because we can test to see whether these antibodies are present. And so we may be standing that up soon. As far as measuring the neutralizing antibody titers, we've been very fortunate that our Canadian National Microbiology Laboratory in Winnipeg had been setting up this neutralizing antibody test obviously for the country for their own use, and they were kindly willing to set this assay up for screening all of the plasma donors. This was important because this assay involves live virus cultured in viro cells, and that means that you have to do this in a level 3 containment facility, which Canadian Blood Services does not have. So we have the NML do this testing. If the antibody titer is at least 1 in 160, then the units are released for clinical trial. And then the other donations that don't have that high a titer, we use them for the production of test panels that are used by laboratories around the country. The next slide, please. So I'm not going to go into the detail here. You probably can't see this anyway, but I just wanted to point out that there are some subtle differences between how we prepare and issue the products from Canadian Blood Services to Héma-Québec to the New York Blood Center for those 3 sites that are providing products in New York, or providing patients rather in New York. And there are just some subtle differences around when we do the antibody testing and how that influences the products we release. But they're basically all convalescent plasma. Everybody has evidence that there are antibodies present. Next slide, please. So I'd just like to say a few things about what we're learning about these antibodies. The first is that the immune response is hugely variable from individual to individual. Most people who get this illness do make an antibody to the spike protein that's fairly high titer, but many of these antibodies are not neutralizing. So there's not a direct correlation between the amount of neutralizing antibody and the amount of antibody to the spike protein. The intensity of the antibody response in a person and how long that lasts is usually correlated with the severity of their infection. But we also have seen some people who have a confirmed RT-PCR infection that have very little antibody. Sometimes it actually lasts for just a couple weeks and goes away, but they've also resolved their symptoms, so something has worked. And although the data sets that are out there to date from various people who are working on this are not really that extensive, it does look like these neutralizing antibodies are not around for that long. So they're around for a few weeks to a few months. And this means that getting a hold of our donors in the right time frame becomes really very important. And so, some of these features of the antibodies may echo back to the talks you heard from last time in this series about vaccine production. Go to the next slide, please. Go to the next slide, Jose. Thank you. So just to show you what the assay actually looks like, down in the lower right-hand corner, this is a plaque neutralization assay. And the little white spots you see are places where the virus is dead, and the blue is where the cells are still, or is everything still alive. And so what we're looking for is a reasonable number of dead virus in these assays. We have a cutoff between 180 and 160, and what we're actually looking to do is to create product that we're pretty sure has got some capacity to kill virus in it. I wanted to just point out in this little box on the left-hand side, this is a study that's just come out within the last couple of weeks as a preprint, non-reviewed, so we'll see where it ultimately ends up. But this was an interesting study done by our colleagues in Seattle, looking at what happened to the antibody levels in various patients that they had collected for convalescent plasma. And one of the things that this very clearly shows is on the x-axis, they're looking at the approximate number of days after the symptoms started, and you can see this fall in the amount of antibody. The other thing that's here is the yellow boxes represent people who were sicker than people in the green boxes, and the blue boxes represent people who were asymptomatic. So these are people who are walking around with viral infection but feeling fine. And one of the important things to point out here is that the sicker people have higher titer antibodies, but also if you just look at the green dots, they're spread over a huge range. And so you have a very wide variation in the response and in the level of antibody that you're collecting from individuals. So this starts to really influence how we can stockpile and collect plasma, not only for clinical trials, but if this works, how do you build an inventory for later? And then the next slide, please. So just to summarize, this is 500 people who got together to do this convalescent plasma trial in Canada. As Jeannie was mentioning, this came about in record time. Lots of sites involved, lots of organizations involved, and fortunately, lots of our fellow Canadians have stepped up to be donors. That last slide. This one just goes in a few stages. If you have had COVID yourself, or you know people who do, who you think might want to participate in this initiative, please help us spread the word. You can go to blood.ca. There's a whole registration process there if you want to be a donor. And I think you can just push through this to the next one. Yeah, so these are just photographs of individuals who've been very actively involved in helping us. And just like to say thank you.
With that, thank you to both Dr. Devine and to Dr. Callum. The best part of being the moderator is the chance to actually learn as you go through your presentations. We've had a fair number of questions raised, so I'm going to kick off with a question that I think, Jeannie, you may have touched on briefly in your presentation. So I think you talked about there being about 53 studies that were underway already. So what do you think the impact of the decision by President Trump on Sunday to push the FDA to approve in its current state with the data regarding efficacy where it is? And there's certainly been a lot of media coverage on it. So what's your expectation of what will happen to those studies at this point in time, if you have an observation?
Yeah, so I think that's a big problem for the scientific agenda on convalescent plasma. You know, many researchers in the United States were had launched, were trying to recruit into these trials. They were already having a problem recruiting patients into clinical trials in the US. And so I think that this is just going to make it harder for them. Many scientists have spoken up about the need for randomized trials to determine both safety and efficacy. And even the FDA guidance document that was released Monday or Sunday states we need randomized clinical trials. So even the FDA is saying that. So I'm hoping that perhaps some organizations in the United States will still be able to, stick to high scientific rigor to get these trials done.
Thanks for that, Dr. Callum. One of the questions, Dr. Devine, was related to what the recruitment numbers look like, and reflecting back on in the early days of HIV and looking for getting people to be involved, that there was concern that you wouldn't find enough patients for these types of controlled trials. Do you have any observations about sort of the supply and your expectations based on where you are right now?
So at the moment, our target, as Dr. Callaman mentioned earlier, was to collect about 1,500 units to start with, and 1,000 of those have come from Quebec, and the Hema-Quebec folks have finished their collections. CBS is looking to collect about another 800, and we're about 500 into that. Our concern is not so much about supporting the trial as it is, if the trial is successful, the demand for this product will start to grow. And so we're actively encouraging Canadians who think they want to be interested in participating in this kind of project to please come forward and register, and we'll contact you and take it from there.
Okay, thank you for that. One of the questions I think probably Dr. Callum for you is just a clarification for those not as familiar with the clinical presentation of COVID-19 at this point. What does the standard of care look like at this point, and then how different does that look with the trial that's underway?
Yeah, so obviously standard of care is going to be different, site by site. Every hospital has a slightly different practice for these patients. So the key things that are being done for most of the patients are attempts to use non-invasive ventilation to try and avoid putting patients on a ventilator for as long as we can, but then also like a safe intubation, giving the patients dexamethasone, giving them a steroid to combat some of that inflammation, using prone positioning to make sure that all lung areas are open. Hydroxychloroquine is not being used. There's variable use of antibiotics to prevent secondary infections. And then there's variable use of, I think everybody's getting prophylactic DVT thrombosis prophylaxis, but some hospitals are actually using full dose because even on prophylaxis, the DVT-PE rate's quite high. And so people are using full dose anticoagulation, and there are also studies ongoing studying that. The two other major clinical trials that are happening in the country are CATCO, which is a drug trial, as well as REMAP-CAP. But our patients can be co-randomized into those studies.
Thank you for that. Dr. Devine, I think this question is for you, and if it's not, please do your tag teaming. One of the questions raised was, how long would development of a hyperimmune globulin, which could be produced from multiple donors, take? So as you're reflecting on what could this potentially look like if successful, and we're needing lots of volunteers, would this help reduce the burden on centers, and might it actually have the ability to have a more potent product?
Yeah, so just for background for the viewers, a hyperimmune product is prepared from pooled plasma from a lot of people who have recovered from COVID, and the antibody molecules are isolated or at least enriched in those products, and then that product is somewhat standardized. So you would have a preparation of a lot, and all the little bottles would have the same material in it. So it's a way to standardize these kinds of antibody treatments, this passive immune therapy, in a way where it's more standardized than what we do if we're just transfusing plasma. So there are clinical trials that are underway for hyperimmune. The large plasma fractionators of the world had gotten together and created what they're referring to as an unbranded hyperimmune. So nobody gets to put their company's label on it, but they're trying to do this to see if they can put something out there that would help COVID patients. So those trials are underway. They haven't completed yet, but we should know in a few months whether that's an appropriate path.
Thank you for that. Dr. Callum, one of the questions raised is for Concor 1, do you allow other treatments such as Remdusifier?
Yes, so we are allowing participation in other clinical trials, and if the physicians wish to use some therapy that they think that may combat the illness, we're just collecting that data so that we can use our statisticians to control for patients that are treated in other clinical trials compared to ours. One of the things that we've done is ensure that we've got a large number of non-academic hospitals enrolling patients. So we have a lot of patients who aren't participating in other clinical trials so that we have some groups of clean patients. But we're going to use our statistical analysis to control for that. We didn't think it was fair for patients to have to choose between one study consent form and another study consent form when they're sick, you know, on oxygen and are short of breath, that was a fair thing to do. And so I think we've made the right decision for that.
Maybe just a quick follow-up to you is on the study itself, and I have to admit, when you were talking about the team, I actually wanted to get involved. It looked like you were having so much fun with this group. One of the questions is, what is your expected timeline for completion of the study at this point, based on what you know?
Yeah, so we're currently at 88 people enrolled. We're consistently getting 1 to 3 patients enrolled per day. And how fast we get enrollment will really depend on people's compliance with masks and social distancing, essentially. And it looks like, you know, the enrollment goes city by city. You know, we'll get a wave in Ottawa, and now, right now, you know, Alberta seems to be enrolling, or then it's Quebec. So I think it's just going to depend on that. We don't look until 600. At 600, we estimate, can we stop? Do we have to recalculate? Is it 800 people we're going to need, or 900, or 1,500 patients to answer the question? And so the big timeline is looking at that 600 mark and having those patients out to their 30-day follow-up.
Thank you. Dan, actually, I'm going to give you 3 questions all combined. They're all along your lines of work, so hopefully relatively simple. I guess the first question is the plasma being frozen? There's also a question about actual ability to donate plasma, and an individual has shared an experience where certain locations in Canada don't do plasma collection, so the example being Victoria or Vancouver. And can you speak a little bit about the availability of being able to do the donation? So maybe, and then I'm not sure which one of you this question is best for. It's with regard to the ABO compatibility, and how is that sorted out, and what was the consideration of arriving at that? So that one might go back to Dr. Callum.
Sure, I can take the first 2. So the question around how it's stored is, yes, the plasma is frozen, generally frozen within 8 hours of collection. That's sort of the typical way we handle plasma. And then the where you can go to donate is absolutely linked to where we have the equipment. So the process involves basically plugging you into a machine that will extract some blood. It spins it in a centrifuge. It gives you your red cells, white cells, and plasma back, or sorry, platelets back, and we keep the plasma. And so it's a minor medical procedure, and we do that under a controlled environment. So it's not something we do on our mobile blood clinics. And so in British Columbia, which is where the question seems to come from, we have plasma site in Vancouver, but we don't have plasma site in Victoria.
Thanks.
And I think I'll turn the ABO question over to Jeannie.
Thanks.
Yeah, so we were worried that we weren't going to have some of those rarer blood groups, particularly the A, B, and the B. But Canadian Blood Services and Hemo-Quebec and the New York Blood Center have, you know, I don't know what they've done to find these donors, but they did. So it really hasn't been a problem. Remember that plasma compatibility is the opposite of red cell compatibility. So AB can go to everybody. A and B can go to O or to A and B. But people that are group O can only get group O. And so obviously it's those AB donors that we would love, because they're, you know, a universal, you know, those donors that can only get AB. And so, but they're rare. It's 4% of our donor base. So if you're AB, you've had COVID, you know, Canada expects you to do your duty and get out there and donate.
Last question, and then I'm going to turn it over to each of you for a closing comment. So last question goes to you, Dr. Devine, which is an interesting question. So since there's so many asymptomatic COVID patients that are never diagnosed, is there any way to test all of the plasma that's being donated under any circumstance to determine if it contains antibodies and therefore potentially expand the donor pool?
Yes, so it's a good question because Canadian Blood Services is actually actively doing that right now. We're testing all blood donors for the presence of antibodies against the COVID-19 virus. And that's being done really to try to get an assessment of what proportion of the Canadian population has been exposed. And so that will pick up people who have had asymptomatic infection and yet have made an antibody. So we've, I think we've reported on, I think the first 10,000 donors that we had tested, but we will continue in collaboration with the Immunity Task Force folks. We will continue to be doing that testing for a number of months.
Thank you. And with that, we're going to wrap up the Q&A session. And thank you for everyone who sent the questions in. I haven't been able to quite get through all of them, but we've certainly captured many of them. So before we go, I'd like to give each of our speakers a moment, perhaps to share if there's only one thing someone has as a takeaway from today's session, what would you want that to be? So I'll go first to you, Dana. What would you want people to remember today?
Yeah, so I think that the take-home message is that this is a promising therapy that we are trying to assess whether or not it actually works, and to please ignore everything you're reading in the media coming out of the U.S.
And the same opportunity for you, Dr. Callum.
Yeah, so I think in some countries, COVID-19 and the medical care of those patients is becoming politicized. Shouldn't be politicized. It's a medical treatment, and we have to listen to the scientists and the science and keep the same rigor that we would normally do for a clinical trial, whether it's a drug trial or a convalescent plasma trial. And let's keep Canada out of this kind of political nightmare, and let's stick to the science. And we've done a great job of social distancing and wearing masks. Now it's time to focus on the science, and let's do these trials. Let's get answers for Canadians.
Thank you for, both for the very important closing remarks. As CADI's role is evidence driven, it's really important for us to continue to create a forum for these kinds of conversations to happen. So a huge thank you to both of you. It's been really heartening to hear the thoughtfulness that's gone into this and the progress and leadership that's being demonstrated here in Canada. Thank you to everyone who's joined the conference today. I think in our registrants, we had individuals register from over 20 countries. So to your last points, continuing to work in this important space is something that we applaud both of you for. On a practical note, as we close off this survey, a short evaluation will pop up in your window. So for all of you individuals who've been listening today, please do take the time to answer the few short questions. It's really helpful to us. And we'll be back in this fall with more COVID-19 webinars, the CADIF lecture series, and other events. So please watch the CADIF website for details, and have a great afternoon. Thank you again to our speakers.
You're welcome.